Okay, so this is our second video on toxicology and human health. Same chapter in Miller, chapter 17, just continuing on. This one will be a little bit shorter, there's not as many slides here. So we're going to focus on here's how we measure toxicity. And then at the end I'll be covering a little bit on what happens to some of these toxins in the environment. So we got to expose species to varying doses and measure the response. And obviously we're going to use, we use all different sorts of organisms for this, but we try to use ones that maybe have a little bit less uh, sentient, a little less conscious. So we use things like flathead minnows and worms and stuff like this. But there's lots of studies that are also done on other animals, higher animals, primates. This is part of the concerns with toxicology studies. But obviously we're not going to test on humans. So what we're going to do is expose them to varying doses and measure response. Now, the response doesn't have to be death. That's just a common thing that's done, is to find that point at which kills at least part of the population. So a threshold dose will be the level below which there's no observable effect. There should be some threshold at which that toxin starts to have a negative effect. Now keep in mind, we said at the beginning, the poison is what makes the toxin. There are some things that are, needed for our human body to function, but at higher levels they're toxic. So everything is potentially toxic just at dosage. So there's different aspects to a curve we can look at. And the curves can look different ways, but this is showing you a common dose response curve. And here you can see there actually is a threshold, a point below which there is no effect. So we typically have a response on the y-axis and the dose on the x-axis. And then you see there's some sort of threshold where we start to see a response. And the no observed effect level is the highest dosage that shows no response. So keep in mind, this is a curve, but it's basically composed probably of individual data points. So there must be your NOEL there, the highest kind of point in which you see no dosage. It's going to be just below your threshold dose, depending on how many data points you have, that type of thing. Obviously up here is 100% response, where we've got whatever that response is. It can be death. It could be things like you could look at reduction in reproductivity. You can look at something biochemical that happens in the body, the production of a particular or depression of a particular hormone or something like that. So the threshold will determine which approach is used to determine harmful effects. And let's jump over to Miller real quick, because he does a real good job here, figure 1714. Here's 1713. This is just a dose response curve, and this one is showing an LD50. So this is the percentage of the population killed by a given dosage. So this is actually, okay, so percentage of the population actually killed. So what you do is you look at what's the dosage at 50% of the population is killed. That's how you determine your LD50. And this might be hypothetical units. It might be something like milligrams of contaminant per kilograms of body weight. That's common. So this is how you would get an LD50. If it's not a lethal dose, we just call it an effective dose, an ED, instead of LD. And that could be an effective dose, a dose at which 50% of the population stops reproducing, for instance. Now, this is the idea. Here we have a case where there's no threshold, meaning we start to see an effect right away. It could be linear or it could be nonlinear. It doesn't have to be not linear. That's a common misconception that it's going to, the higher the doses, the greater the effect. No, here you can see you have a slow start, very little response early, and then it starts to pick up. This one is linear, straight out the same. Increase the dose by double the dose, you're going to double the effect. So scientists used, I'm reading this over here, two types of dose response curves to help them estimate the toxicity of various chemicals. The linear and nonlinear curves in the left graph apply even if the smallest dosage of a chemical has a harmful effect that increases with the dosage. The linear and nonlinear curves in the left graph apply in even the smallest dosage of a chemical. Yeah, that's poorly worded actually, unless I'm reading it wrong. The linear and nonlinear curves in the left graph apply if even the smallest dosage of a chemical has a harmful effect that increases with the dosage. Okay. The curve on the right applies if a harmful effect occurs only when the dosage exceeds a certain threshold level. Which model is better for measuring the effects of a specific harmful agent is uncertain and controversial because of the difficulty in estimating the response at very low dosage. That's true. And that is the gray area of toxicology. What happens at really low doses? It's something we don't have a lot of data on. Typically, carcinogens, we use this type of approach and model over here, is we assume, because they're carcinogens, we assume there's an effect at all doses, which is not necessarily the case. Most carcinogen models would say actually that's not the case. But it's kind of like a safety thing. We're not going to take any chances sort of thing. All right, jump back to the slides. Okay, so 50% response is a common target. Things are changing in the toxicology field. They're now looking at lower response rates, so you harm less animals. So we can look at like a 10% response rate. You just want to make sure that you get enough response that you have statistical significance and meaning to it. And there's where I said if it's lethal or not. So, question to you, how does size or weight affect this? Okay, so that's something I want you to think about. I mean, I have the answer here. Dose is usually reported as milligram per kilogram, and milligrams of toxin per kilogram of body weight. So think about why. Obviously, why weight is important. And we could talk about that at our Zoom sessions. So acute versus chronic, these are good types of things to get down for quizzes, basic definitions. What's the difference between acute and a chronic? Acute is short term. Could be shorter than weeks even, but usually no more than weeks to maybe a month or two. Chronic is long-term years. So, if you were to drink water that has a trace amount of chemical in it over the course of 10 years and you develop cancer later on, that would be a chronic effect. Acute effect, hey, you start drinking this water, in a few days you start developing a rash on your skin, that's acute. So these are just some LD50 values just to give you some ideas. So look at DDT, because we've talked about DDT enough. And it's 100 milligrams per kilogram of body weight. So it's not particularly toxic compared to some things. I mean, nicotine is more toxic. Arsenic's more toxic, almost more than twice as toxic. There's caffeine, just to give you an idea. Sugar, okay, has an LD50. Like we said, the poison is the dose. Remember that. And this is for rats or mice. This is for how these were tested. So just to give you some sense. So our interest is mainly humans, so the question becomes how reliable are animal tests? Well, to give you an idea, Dioxin Toxicity, the LD50 for guinea pig is this. Now that's 0.0006 milligrams per kilogram, or if you want, let's multiply by 1,000, 10, 100,000, 0.6 micrograms per kilogram. I just multiplied it by 1,000. But for a hamster, it's 1.1 milligrams per kilogram. This is more than 1,000-fold less toxic. And what's the difference between a guinea pig and a hamster, right? You would expect these to be similar, and they're not. So the question becomes, how toxic is it in humans? This is one of the problems with doing animal toxicity. We really don't know. So when dioxin first came out, the initial studies were done with this, and people were saying this is one of the most toxic substances we've ever known. And then as they did more studies, they realized, well, wait, this is much more biochemically specific. And that's the key, is that even though our systems are generally the same, there's lots of differences in receptors and different proteins and different enzymes and different binding sites. So that's the key, not the bulk systems, and that's where you see different effects. And keep in mind that most of these toxicity effects we're talking about, are kind of non-target effects. Especially when you talk about a pesticide, we'll see that. It's not what the pesticide's originally designed to do to the insect. It does something else in our bodies that causes problems. The other thing to think about, a couple good definitions to know for quizzes, is synergistic and antagonistic effects. We usually study one chemical at a time because, just like you're learning as you do your lab projects at home, the more variables you have, the more difficult it is to come to conclusions. So you study one thing at a time. Well, we should know that we're often going to be exposed to a group of chemicals rather than a single one. Synergistic effects is when that you have a greater effect. And you can even have less effect because they can interfere with each other. So let's practice just some basic kind of conversion metric system related to dosage and stuff. If the concentration of a toxin in drinking water is 5.5 micrograms per liter, what's the concentration in nanograms? Now, if you remember, micro is 10 to the minus 6, nano's below it, 10 to the minus 9. So remember, if you go down, the number should get larger. That's what I taught you. And that's what we see. We go from 5.5 to 5.5 times 10 to the third. There's the conversion. They're a difference of a factor of 3 apart. So there's 10 to the 3 micrograms in a nanogram. Or your brains might work better like this. 10 to the 3 nanograms in 1 microgram. You get the same answer. Working with negative exponents sometimes is a little more confusing to people. They're 1,000 apart, so you can always say, it's just like saying there's 100 pennies in a dollar. No one says there's $0.01 in a penny. So same thing. Realize that there's more of these within that. So this is like the penny here, and this is the dollar. And you get the same answer. Realize, unit-wise, what we're doing, we're canceling out the micrograms, and we're getting nanograms. And our volume is still there, so we still have nanograms per liter. We were asked for microliters, so we still have to convert it. So we now have this 5.5 times 10 to the third nanograms per liter, and there's 10 to the minus 6 liters in a microliter, or if you will, 1 liter is 10 to the 6th microliters. Both answers are the same. Either way you think about it. So this is your final answer that you want here. I forgot that I asked you also to change the volume. Just a way of showing you how to convert. We're going to be doing a lab on dilution and pollution. Dilution is not the solution to pollution. Whether you've done the lab already or are going to do it, this is similar. All right. Now let's look at determining LD50. Average cup of coffee, regular coffee, contains about 100 milligrams of caffeine. And the LD50 for caffeine for rats and mice is 130 milligrams per kilogram. We'll have to use that for humans. Assuming that the LD50 for humans is the same, determine the number of cups of coffee that each person in a group of 150 adults would have to drink at one time for the caffeine to be fatal to 50% of the group. So the idea is we're trying to see what that LD50 is, okay? We know what the LD50 is, so we're trying to see, okay, well, how many cups of coffee is related to that? So you should think about this and solve it, try to solve it. I think I'd rather do this in person than doing it now. I'll explain it, but here's it solved. I will give you the conversion. This is the conversion that I'll give you. Okay, 2.2 pounds in 1 kilogram. So we now know this person weighs 68.2 kilograms. And we know you need 130 milligrams, right, per kilogram. And we know we got 100 milligrams. So let's just figure out how many cups of coffee, basically, right? That's all we're doing. So that's how many kilograms the person weighs. You need 130 milligrams per kilogram. There's the 8860 milligrams that you need to kill 50% of the population. And since 1 cup is 100 milligrams, we wind up with 88.6 cups. So I went through that pretty fast because I want to talk about it during our Zoom session. So give yourself a chance to look that over. Make sure you can understand that and solve that type of simple LD50 type of calculation, and we'll talk about that. And these little hours, these little lines just showing you unit conversions. Now, I want to talk about what happens to these toxins in the environment, how they stick around, and it can be, some are broken down quickly, some are not. We're going to introduce half-life again, which we use for radioactive decay, but it's the same thing here. So persistence tells us toxic levels can build up. They can last a long time. That means under given conditions, they don't break down. And we already know half-life is the time for half of a substance to decay. So it continues repeatedly, as we know. So for DDT, that half-life is 10 years, which means after 20 years, after 10 years, you got 50% remaining. Take half of that, 25%. After 20 years, there's still 25% of the DDT remaining in the soil. Okay? That's specifically for soil. Obviously, it'd be different in water. So remember, fraction remaining is 1/2 to the n, so n is the number of half-lives. So the question remains, how much DDT remains after 40 years? Well, how many half-lives is that? Well, if the half-life is 10 years, just 40 divided by 10 tells me 4 half-lives. So it's 1 half times 1 half is a quarter, times 1 half is an eighth, times 1 half is a 16th. And a 16th, if you did it on a calculator, 1 divided by 16, you get 0.0625, or 6.25%. Now, if you're asked how much remains and you were giving a starting amount, 500 milligrams of DDT, how much remains after 40 years? Well, then just simply multiply that 0.0625 times that starting amount, and you have the actual amount that remains. So think about that one too, and ask any questions during the Zoom session. And this, once again, is just showing you a scale of half-life. I did this already for radioactive decay, so I'm not going to cover this here. Now, as we go into breakdown of chemicals, what can happen? 3 main things: hydrolysis, reaction with water; photolysis, reaction with light; and breakdown by microorganisms. There is also chemical reactions that can happen that are different than hydrolysis or photolysis. We call these oxidation reduction reactions. I talked about it in the renewable energy section with fuel cells. Just realize that there are other things other than hydrolysis UV light. You can have species in the soil that actually can attack the chemical, like iron or manganese or something like that, some elements that will react and break it down. This is specifically with microorganism biodegradation. Movement of toxins. So one thing you learn kind of early on, maybe even middle school, is like dissolves like. That's why oil and water don't mix. They're kind of not like each other. Okay? And what I say to my students is look for pieces of water. When I say pieces of water, I mean if you take H2O, that means this, okay, or just the oxygens. The best is the OH. The bigger the piece, the better. So this is very soluble in water. That's just methanol. And ethanol is, which is the alcohol in alcoholic beverage, is very soluble in water. Because you got a piece of water there that helps it interact with the water. Now, something like octane, that's 4, 6, 5, 6, 7, 8. That's octane, just a straight hydrocarbon with carbons and hydrogens. No pieces of water. Hydrogens don't count. No pieces of water, doesn't mix well with water. Oil and water don't mix, and that's basically gas, right? Now, I could put an OH group on here. And what I want you to realize is that that makes it more soluble. But it's a pretty small piece considering the size of the molecule. But if I then go ahead and make this a 2 here and put another OH here, that makes it even more soluble. And I make this 1 hydrogen here and put another OH here, that makes it, you see, get the idea, the bigger the molecule, the more pieces of water you need. This approach is something I developed, and allows me to not talk about hydrogen bonding and electronegativity and dipoles, all the stuff my chemistry students need to know to understand that. So when you look at something like glucose, which is a sugar which has a bunch of OH groups on it, you realize sugar goes into water very easily. Now, that doesn't explain why something like this is soluble in water, sodium chloride. That's because this has charges. So if it doesn't have OH groups, it has to have charges like this. And that's an ionic compound that has a metal and a nonmetal. These are going to be soluble in some cases. Not always, because some of them sometimes bind really strongly. And I don't expect you to know this. I want you to be just be aware of it. Something like lead sulfide. That's lead 2 plus and S2 minus bound together. These are so strongly bound that they don't dissolve in water very much at all. And that's not something you would ever know. I'm not going to go over solubility guidelines with you the way my chemistry students are. I just want you to realize that, hey, if it doesn't have a piece of water, it can still be soluble if it's one of these soluble salts. But there are, be aware of, insoluble salts. Okay, so that's why oil and water don't mix. That's why ethanol and water do mix well. So if it's a water-soluble toxin, it spreads very rapidly. It gets in the water, dissolves, and can spread. Groundwater, well water, surface water. It does become diluted because it's dissolving in the water. And it typically, not always, but this is a generalization, but in general, broken down easier. Why? Well, because you got reactions like hydrolysis, which require water. That's what this means. Hydrolysis is reaction with water. Also, microorganisms live in water films, so they like water films if they're in the soil and stuff, and that's going to make it more subject to biodegradation. It's not a definitive rule. There are some things that are water soluble that break down less readily, and some things that sometimes aren't water soluble that can break down easy, but that exception is even less. Where fat soluble toxins, they have poor movement. They have to spread by some carrier other than water. So imagine you have water flowing and you have a bunch of soil particles and sediment particles that are dissolved and flowing. Well, they can carry the contaminants. They tend to be more persistent, so they don't break down as easy. And they tend to concentrate in fats and oils. So if they get into an organism, they concentrate in the fats and oils, and then we don't metabolize them, and then they just collect in the body. That's called bioaccumulation. And if it moves through the food chain, it's called biomagnification. There's actually another term, bioconcentration, but in general, fat-soluble toxins collect in tissues of aquatic organisms. Bioaccumulation can magnify in the food chain as organisms feed on each other, biomagnification. And they collect at concentrations that are higher than the surrounding water. That's actually what bioaccumulation means. I'm sorry. Bioaccumulation just means they collect in the tissues. That's fine. It concentrates in the tissues. Often you find a concentration higher. DDT does this. So here you would see concentration that's higher than the surrounding water. That's the bioconcentration. Accumulation is just accumulating in the organism, and magnification is getting more and more through the food chain. This is what happened with DDT and the birds of prey and the raptors and all of that led to the banning of these organochlorine insecticides and phasing out of those compounds for different types that aren't as persistent and don't bioaccumulate. So here you can see, this is not from your book, but this is measurements in the plankton. Down here, it's only at 0.04, and this is units of parts per million, so milligrams per kilogram of fish weight. In the clams, it was up to 10 times more, 0.42. In the fish, not quite 10 times more, about 5 times more. And then into the fish-eating birds, heck, up to, 15 times more? Or no, up to 30 times, 35 times more, roughly, potentially as high. So there's a big range here. You see it here. This is the age of the trout, and the longer it lives, the more accumulation. This tells you that it's not being metabolized. It's just collecting in the body. So this is EPA's persistent criteria for toxic chemicals. By the way, we're almost done, so we'll keep this under 30 minutes. So with air, it's very simple. If it's greater than 2 days, it's considered persistent, highly persistent. There is no middle ground there. The others are all the same. Less than 2 months is non-persistent. Greater than 2 months is persistent. Greater than 6 months is highly persistent. And these are examples of persistent organopesticides that have been banned, and these are all insecticides. And this, of course, is DDT that you're all familiar with. But these are all very similar, and you'll notice no pieces of water, right? This one has one little O group here. And also, when you see these, group 7, the halogens, that increases persistence. And the more there are, the worse. Biological systems don't deal with a lot of halogenated substances. That's just not common in biological systems. These are man-made, and they're heavily halogenated. And that's not common, although some stuff is produced naturally in our biosphere that is halogenated. It's not real common, and heavy halogenated is even less common. And when you see that, you tend to think more persistence. So DDT was banned. DDT concentration increase of 10 million times in this case, from this concentration in the water to this concentration in birds. And what we found out that DDT interferes with calcium metabolism. I used Ca2+ because that's what's used to build the eggshell. It's calcium ion. Remember I talked about the ions are the ones that are biologically used and active. It thinned the shell, so when they would be born, a lot of them wouldn't survive. So it was banned due to this persistence, bioaccumulation, and toxicity. It's designed as an insecticide, so it goes after the nervous system in the insects. This is what we call a non-target effect. That's not the way it was designed to do, what it was designed to do. So it doesn't affect our nervous system the way it does with an insect because the concentration's too low. But as it collects in the organism, it's interfering with other things. So these are just some toxic organics. These are used in transformers, so in electrical grid systems. So there's little green boxes that are on people's lawns. This is where you would find some of these, or they've been phased out. They've been banned in this country. Dioxin is not something we produce. This is only formed in fires, high-temperature fires that include biological materials because biological materials has carbon and it has chlorine. Chlorine's pretty common in just as the ion form because you know salts, sodium chloride, pretty common in biological systems. So the presence of two at high temperature forms dioxins. They'll form in incinerators, forest fires, things like that. I don't believe they form in household fire pits and fireplaces, or they might, just depends on the temperature. These are known as organochlorines. So this is a basic summary then of what we covered. Basics are dose, response, types of toxins and examples. So that would be from the first part. And then the fate of toxins at the end. So this is not in the order of the slides. It's just kind of highlighting some of the important things. The basics would be kind of how we measure toxicity. Then there's types of toxins and their effects. Then there's carcinogens, mutagens, teratogens. I put those all together. And then fate of toxins at the very end. And that's basically your video and the end of your toxicology human health videos because that's the second one.